Are you ready?
Good day everyone and welcome to our video presentation. It is an honor for us to be able to share information to such a great audience. We are here today to provide you some exciting information about varieties of local and international published research related to marine fisheries, which is very beneficial for you in the future. But before proceeding, I'll ask you this. Have you ever thought how the world might have been without any technological advancements? Well, the life that people now have, or the things that we can achieve in minutes that previously seemed unattainable, are all because of research. Research is the finest and most dependable way to comprehend and act on the complexities of various issues that we as humans are facing. Without a further ado, we will now first discuss the 5 international published research. Research title: Lipid membranes modulate the activity of RNA through sequence-dependent interactions. The author of the research is Thomas Zerniak and James P. Saenz. It was published on January 18, 2022. The instrument used is experimental instrument wherein RNA incubations were prepared. RNA concentrations was determined by measuring absorbance and performing a denature page analysis. They conducted their study at Public University in Dresden, Germany. The DOI number is presented on the screen. Next is the analysis on the strong parts of the research and why. Lipids, which are essential for life in one of the most ancient biomolecules, can spontaneously self-assemble to form membranous bilayers, theoretically providing a surface that can serve to concentrate, protect, and regulate RNAs. It shows that direct RNA-lipid interactions can modulate ribozyme activity in a lipid-dependent manner. It further demonstrates that RNA-lipid binding is influenced by nucleotide content and base pairing of RNAs. This yields an essential framework for engineering RNA lipid system that can be regulated based on sequence specificity and introduces a mechanisms for riboregulation in cellular, synthetic, and prebiotic systems. Next is the analysis of the weak part of the research. The scientists tested how well different types of RNA molecules interacted with lipid membranes. They found that some RNAs were binding lipids better than others, and that depended on the sequence and structure of the RNA molecule. Guanine, one of the 4 building blocks that makes up RNA, was crucial for RNA to stick to the lipids. Adding additional chains of guanines to the RNAs made them even stickier, providing a way to control the strength of RNA-lipid interactions. In particular, the presence of guanine in short RNAs is crucial for RNA-lipid interactions, and G quadruplex formation further promotes lipid binding.
Another published international research is entitled Lipid-Lowering Effect of Lotus Leaf Alcoholic Extract on Serum, Hepatopancreas, and Muscle of Juvenile Grass Carp via Gene Expression. The authors of this research was Junping Yao, Pingqing Ho, Yanhong Zhu, Yingyan Su, Qingsun Tan, and Sufang Liang. It was published on December 4, 2020. The methods of this research is experimental. This research was conducted in the College of Fisheries, Guangzhou Agricultural University, K Laboratory of Freshwater Animal Breeding, Ministry of Agriculture, China, Hubei Provincial Engineering Laboratory for Pond Aquaculture, Engineering Research Center of Green Development for Conventional Aquatic Biological Industry in the Yangtze River Economic Belt, Ministry of Education, Wuhan, China. The OI number is on the screen. The strong points of this research is lipid accounts for most fat present in the body. Lotus leaf prevents fat accumulation in human body and may have similar effects on fish. In intensive aquaculture of grass carp, fish display disordered lipid metabolism, showing excess lipid deposition in the hepatopancreas and muscle. This results in physiological impairment and degradation of flesh quality. All of this are caused by formulated feeds which contain lipids. The study aims to investigate the potential of lotus leaf in preventing the accumulation of lipids in the hepatopancreas, muscle, and serum of grass carp. In this research, the researchers were able to decrease lipids in the said fish tissues. Using the 4 experimental diets of 4 groups of alcoholic extract lotus leaf, namely lotus leaf alcoholic extract 0 or the control, lotus leaf alcoholic extract 7, lotus leaf alcoholic extract 14, lotus leaf alcoholic extract 21. In this formulation, the researcher was able to conclude that lotus leaf alcoholic extract decreased the triglycerol in the serum, and on the oil red O stained hepatopancreas and muscle sections, lipids content decreased as dietary lotus leaf alcoholic extract level increased. The weak points of this research is the researchers tried 4 groups of lotus leaf alcoholic extract formulation with different levels in the experiment and found that not all of the lotus leaf alcoholic extract group has the same effect when it comes to lowering lipids in hepatopancreas, muscle, and serum. The effect is dependent to the levels of formulation. Good example was the effect in the serum, where cholesterol content in the lotus leaf alcoholic extract 14 and lotus leaf alcoholic extract 21 was lower than in the control and lotus leaf alcoholic extract 7. The higher density lipoprotein content in lotus leaf alcoholic extract 21 is higher than in the other group. The lower density lipoprotein content in lotus leaf alcoholic extract 14 and lotus leaf alcoholic extract 21 was lower than the control group and the lotus leaf alcoholic extract 7.
The title of this research is Effects of Dietary Lipid Source and Level on Growth Performance, Blood Parameters, and Flesh Quality of Sub-Adult Olive Flounder or Paralichthys olivaceus. So this is authored by Dong-Kyu Kim, Kyung-Dak Kim, Jo-Young Soo, and Sang-Min Lee, which this research is published on June 2012, and this study is conducted in a fish farm in Olgin, Korea. They uses experimental and observational instrument, and the DOI in this research is 10.5713/AJAS.2011.11470. The strong points in this research is that they organize the result well and provide tables to help the reader to understand the research more. Their research also came up with the result that fish oil in fish meal-based diets for sub-adult olive flounder could be replaced by soybean and linseed oils without negative effects on growth and feed utilization, which is a good thing. They also really show how the dietary lipid source affect the level on growth performance, blood parameters, and flesh quality of sub-adult olive flounder or the Paralichthys olivaceus. The weak point in this research is it is time-consuming, and they also did not include or disregard other factors that can affect the result like parasites, bacteria, and others. There could also be human error because they have a lot of computation and observation to do as well as the extraneous variables that they cannot always be controlled.
In international research, we have a variation of lipid and fatty acids of the Japanese freshwater eel, and the scientific name Ungula japonica, during spawning migration. The author of this study is Haruki Saito, Haruki Kurugi, Shinen Zhou, and Ritaka Michioka. It was published in June 2015. The study conducted at Ishikawa Prefectural University. Study method, sampling, and optimization, and the DOI number were found in the screen. And in the strong statement analysis, the researcher studied lipid and fatty acids composition of the muscle of the wild Japanese freshwater eel. It was analyzed between the initial and terminal stages of spawning migration in order to clarify the relationship between lipid physiology and maturation. The researchers found that an Japanese freshwater eel have a high in 6-FOFA, also known as polyunsaturated fatty acids. It indicates that an Ungula japonica may use this important energy for spawning and before ending its life. And in the weak part analysis statement, the chemical composition of fish can be affected by environmental condition such as feeding condition, season, and sexual maturity. Considering that Ungula eels depend mostly on aquatic benthic animals and small fish, many of which differ with habitat environment, the chemical composition of Ungula japonica may be affected by season and geography.
Hi everyone, so this is one of our international researches that we found. So the title of this research is the roles of lipids and nucleic acids in HIV-1 assembly. So the authors of this study or research is Aina Afadli and Eric Barkless, and it was also published on May 28, 2014. The study takes place in the Department of Molecular Microbiology and Immunology, Oregon Health and Sciences, University Portland, USA. It is also a laboratory experiment. So the research shows the structural aspects of retroviral matrix proteins, retroviral MA binding to nucleic acids, the role of MA binding to nucleic acid binding, and also the inhibition of HIV-1 mRNA binding, which explains the roles of lipids and nucleic acids in HIV-1 assembly. Well, the weak part of this research is that the authors declare that the research was conducted in the absence of any commercial or financial relationship that could be construed as a potential conflict of interest.
Another published local research is entitled Mineral Availability, Dietary Fiber Contents, and Short-Chain Fatty Acid Fermentation, Products of Color Palentelefera and Capaphycos alvarezi seaweed. The authors of this research was James David S. Alcantara and Nancy Lazarolianos. It was published on May 12, 2020, and this research used experimental method, and this research was conducted in the Department of Science and Technology, Food and Nutrition Research Institute, General Santos Avenue, Bicutan, Taguig City, Philippines, Chemistry Department, De La Salle University, Manila, 2401 Taft Avenue, Manila, Philippines. The strong points of this research is the researcher was able to present on which among the two samples are contains the higher mineral content and high availability, and which among the two contains higher soluble dietary fiber, which can prevent chronic diseases. Both samples also produce propionate SC fast. This production of propionate suggests that these two samples have the potential in preventing cardiovascular diseases. Lastly, both samples showed low fat content, which indicates that the two seaweeds have a less than 4% fat and are considered as not good source of crude leaf. The weak points of this research is Decapaphycos alvarize, found to have produced both propionate and butyrate AC fast. However, potential presence of contaminants like heavy metals in the sample must be investigated for safety assessment. It also recommended to analyze the presence of other chelators that might affect mineral availability. Further studies are also needed to validate the physiological functions of the dietary fiber and short-chain fatty acid content, both in selected samples.
Another local research is entitled Fatty Acids in 6 Small Pelagic Fish Species and Their Crustacean Prey from Mindanao Sea, Southern Philippines. So, this research is authored by Ephraim Bicoy Methelio and Anna Arlene Asperas Iya, which published in August 2014. So, they conducted this study in the Pulot City, Mindanao Sea, Southern Philippines, and uses experimental and observational instrument. So it has no DOI, and the strong points in this research is they have very detailed information about the experiment on how the researchers conducted and collected the sample. They also provide tables and diagrams to show the result, which can help the reader to understand the research more. The researchers also made strong points in deducing the trophic linkage among species in support of the use of fatty acid as trophic biomarkers. The weak points in this research is that they lack recommendations of their research for the future use, and they have many species involved in the experiment that affected the result, and they encountered difference due to metabolic differences between cold and warm species. It also involves a lot of analysis, technology, and substances to be used.
So hi guys, it's also one of the local researches that we found. So it is entitled Critical Problems Associated with Climate Change, a Systematic Review and Meta-Analysis of Philippine Fisheries Research. So this research was published in August 3, 2021. The authors of this research is Jimmar C, Elaine Cunatana Baroza, and Kay Ilyushan Munoz. Environmental Science and Pollution Research. This paper proposes to analyze the scientific production of climate change and fisheries in the Philippine context. This research was chosen considering the continuous increase in scientific studies related to climate change and fisheries, and will therefore help in directing researches on future directions for research to aid in addressing critical issues in the Philippine fisheries. The contribution of this paper is to call for both public and private sectors by responding to the management strategies and policies changes in which address the gaps mentioned in each main theme. The economic spheres of fishing with respect to livelihood generation should be anchored on the ability of fisheries sector to cope with environmental changes. Cooperation and integration towards decision making are vital, as pointed out in the second theme. Capacity building and human capital investment can hence ensure that no specific group gets left behind and marginalized in the development of fishing. There should be recognition of the role of women in the fisheries sector and demands for equal opportunity. So that's all for the local research that of this paper.
The next local research was entitled Influence of Different Extraction Methods on Fatty Acid Composition of Lipid Extracts of Chlorella vulgaris Bejering from Laguna de Bay, Philippines. So this is authored by Rochelle Joey A. Saracanlao, June Owen Nacorda, and Hidilisa P. Hernandez. And this study is published on August 7, 2018, and they get the sample in Laguna de Bay and maintain it in the UPLB Museum of Natural History Algal Culture Collection in the Phycology Laboratory 1 of the Institute of Biological Sciences, College of Arts and Sciences in University of the Philippines in Los Baños. So they use this experimental instrument and this study has no DOI. The strong point in this research is that they showed good information about how the research focused on the cultivation of algae for lipid production, specifically biodiesel via lipid transesterification, and how extraction procedure along with the solvent system affects both yield and lipid profile. The research paper also provides pictures, diagram, and charts for the result and discussion, which is a good part to easily organize and explain the result.
The weak parts of the research are the following. It's uncertain for errors. The scope of the study is only limited. And it only shows or introduces few extraction methods. The extraction procedures are incomprehensible, especially to the pioneers.
The title of this study is DNA barcoding of commercially important grouper species Perciform sirenidae in the Philippines. So this is authored by Simon G. Alcantara and Apolinario V. Yambut and been published on September 19, 2014. So this study is conducted on Biotechnology and Analytical Laboratory, Central Luzon State University, Science City of Muñoz, Nueva Ecija, Philippines, Molecular Biology and Biotechnology Laboratory, College of Fisheries, Central Luzon State University, Science City of Muñoz, Nueva Ecija, Philippines, and Freshwater Aquaculture Center, Central Luzon State University, Science City of Muñoz, Nueva Ecija, Philippines. They use experimental instrument and the DOI of this study is 10.1740.1736.2014.958672. The strong part in this research is, aside from reinforcing the classical methodology of grouper identification in the country, this pioneering study on molecular identification of Philippine groupers constitutes a significant contribution to the DNA barcode library of the Philippine marine fishes and to the global barcode entries in general, which can be used when dealing with grouper taxonomy, biodiversity, stock assessment, and trade. The results reveal the different localities where the grouper species can be possibly sourced out in the country for trade and aquaculture purposes. Several of the grouper species are included in the IUCN Red List of Threatened Species as a tool for conservation ecology. And this study signals the implementation of sustainable fisheries management regulation to protect in particular those which are listed under the IUCN.
For the last part of our video presentation, we will be sharing our thoughts on what is our future plan on our research or our research interest and why.
In my personal monologue, so as a BA specialist student, we should able to study the marine biodiversity. We should also protect our ocean, at the same time study more and so that discover species that can be cultured. So it could be difficult for me to create a research title, but my interest studies in the future will be something about fish growth of certain species. So that will be all, and thank you.
Being a fishery student, it is my duty to help and contribute to the Philippine fisheries industry. Studying in this field of science, it surprised me because on how this field plays an important role in the environment and sewage in food security. We cannot deny that our population is continuously expanding, and with this expansion, it also increases the demand for food consumption. And according to statistics in 2019, estimated 87 million Filipinos eat fish with per capita consumption at 35.2 kilograms per year. If this per capita consumption of millions of Filipinos will only rely on capture fisheries, then it's impossible to sufficiently supply these numbers of people. Well, fisheries were already found a solution in this problem. They've established and introduced aquaculture to have a sufficient supply of fishery products to the market. aquaculture, able to have unseasonal production, which really helped the market demand for aquatic products. Today, there are numbers of extensive and intensive aquaculture all over the world, specifically here in the Philippines, and those aquaculture companies aim to quickly produce a marketable sized and good quality of fishes and to gain more profit. The things to consider to be able to produce a marketable sized of cultured organism in just a short period of time is the quality of diet which being provided to the cultured species. High nutritional types of feeds must be used in aquaculture to achieve high production. However, highly synthetic aquaculture feeds might harm the environment because of its chemical formulation and content. With those facts, I am very interested to conduct research and to formulate and create nutritional feeds for aquaculture species. I will formulate a feed coming purely from natural resources will only have a few chemical compositions, not pure synthetic, but high in nutritional quality. Through the guidance of the things I learned in my recent subjects, I think I can formulate and produce high quality and eco-friendly fish feeds. Fisheries taught me to not only think about profit, but also to look what is best for the environment. Aim for good production, at the same time, preserve the environment.
My tentative research, entitled Modern Scientific Medicine to Terminate Fish Diseases. Diseases of fish can be important because they may induce mortality. Sublethal diseases may cause poor growth conversion, poor flesh quality, or undesirable visual changes. Furthermore, some pathogens of fish are infectious to humans.
In the future, I want to do a research about the life cycle of a certain aquaculture species and how they reproduce in order to know what to do to increase their productivity rate. So, I am very interested in helping the fishermen to be attached in aquaculture because I believe that aquaculture can help solve the overfishing issues in our country.
And also for my future research plan, I think I would be more focused in marine fisheries here in Awan, or just the climate change does affect marine fisheries here in Awan, something like that. I would like to have a research just around here in our province, and that's all.
If I will be making a research, the research title will be all about the ocean pollution and the fishing industry problem solution. Because water contamination is a human concern that threatens the lives and livelihood of many people all over the world. Rapid population growth, combined with growing industrial and technical progress, has resulted in severe contamination of available water sources. While ocean waters cannot be taken directly by humans, they do provide a home for many fish species and marine life that are on the verge of extinction due mainly to pollution. This remark raises the most important inquiries. What is pollution in the ocean? What causes this? What effects does ocean pollution have on humans and marine life? What solutions are available to address this problem? What recommendations may be put in place to supplement the remedies in order to discover long-term solution to this problem?